Hello, I'm Nadine.
I'm Nestor.
We are from Team Synthetica Rwanda and we are submitting this project to Goge Competitions 2024.
Our project title is on detection of microplastics using protein-based bioreporter. So Nadine, tell us briefly what are microplastics and what are their effects to human health?
So microplastics are particles that are less than 5 millimeters in diameter and they are mainly spread in the water ecosystems. They come from the degradation of other plastics and they have a negative impact on human health and on the ecosystem since they are bioincompatible and they accumulate in the ecosystems. So the recent research on microplastics is still lacking as it does not contain much information on how it can affect human health. So Nestor, there are detection methods that are used. Can you tell us about them and what they are lacking?
Currently, in detection of microplastics, the methods which are used are visual inspection, Fourier transform, infrared spectroscopy, to name a few. And these methods relies on detection of chemical compounds like functional groups present in these microplastics. They are labor intensive and not suitable for the on-site analysis science. Uh, Samples are taken from the site and analyzed in a laboratory.
So what are the solutions or what is the solution that is proposed by our project?
Our project focuses on production of a bioreporter for qualitative detection of PET plastics. This bioreporter utilizes a PETase enzyme affinity to PET plastics and also a GFP as a signaling molecule. And Nadine, could you tell us how our bioreporter is different from other current methods which are being used?
Yes, so this bioreporter will offer a quick and on-site detection solution in aquatic environments. As we said, the other methods does not do an on-site detection, so this will come as a solution. And it is also cost-effective and can aid in early diagnosis and prevention of these microplastics. Now let's move on to the next part, which is the experimental design of creating the bioreporter, which will be made by the PETase enzyme and green fluorescence protein to detect the microplastic presence. So as we've been saying, the goal of this experiment is to create a bioreporter based on the PETase enzyme and GFP, which will be used to detect the microplastic presence. An expression cassette will be designed and then it will be inserted into the PET11A and CYP119 plasmid. Yes, so Nestor, can you tell us about the expression cassette? What is it and how this be designed?
Thank you, Nadine. As our experiment is on production of a bioreporter which is made of a GFP and PETase enzymes fused together to produce a signal which will indicate microplastics present in aquatic environments. The expression cassette that we'll design will contain a T7 promoter, an RBS34, and a PETase with the histoc, a PETase gene, of course. And the promoter will be induced now by IPTG, the inducer on LAC operator on an T7 promoter. This IPTG stands for isopropyl beta D1 thiogalactopyranoside, and this will induce expression of this gene, be translated into a reporter protein, which is a PETase protein. Now, after transcription of a reporter protein, this protein will be fused with a GFP, green fluorescence protein, by a linker sequence. And after fusion of this GFP and PETase, this will form a PETase GFP complex, and this complex will manifest itself as the PETase will encounter microplastic. This PETase will degrade this microplastic as well as a production of a signal from GFP signs. They are fused together. So it will act as a fluorescent tag which will be manifest, which will produce a signal when PETase degrade this microplastic. Now, after designing our expression cassette, Nadine, could you tell us the next following steps?
Yes, so the next step will be the recombinant DNA technology, where the cassette will be inserted into this plasmid, the PET11A and CPY119 plasmid, and then this plasmid will then be inserted in the E. coli, and then the E. coli to confirm for sure that the cassette has been inserted, the E. coli will then be cultured on the ampicillin to like test the resistance, and those that has resisted to the ampicillin, since this plasmid contains the ampicillin resistance gene, so those that have resisted will then be selected, since we are now certain that they contain the cassette. Or another method that can be used is using the PCR with then the specific primers that are specific to the gene that we have inserted. So that is how, that is the next step that we'll be following after designing the cassette. So now that we have inserted the plasmid in the E. coli, what follows? Like what happens inside the E. coli after the insertion of that plasmid?
Thank you. When the plasmid is inserted into the bacteria, the plasmid which contains our expression cassette, now it will be integrated into the nucleus of this bacteria, and now this gene expression will happen through the induction of this plasmid by an IPTG, isopropyl beta D1 thiogalactopyranoside, and to the T7 LAC operator. And then the process will occur by the binding of IPTG to the LAC repressor, allowing RNA polymerase to transcribe the gene of interest, which now contains our expression cassette we have designed. So after now making a transformation of our expression cassette in E. coli, now what follows when we are going to detect the microplastic presence in samples?
Yes, so now that we have our E. coli that have the ability to detect the presence of these microplastics, we will now culture them, and then the broth culture will be exposed to the collected amount of microplastics. That means the sample that we suspect that contains microplastics. And then when that sample contains microplastics, fluorescence protein will then be observed in the samples. This will be caused by the green fluorescent protein, so that means that a green color or a green fluorescence will be observed in the samples that contains the microplastics. So now, can you tell us about the materials? What materials will we use, and then what are the methods that will actually be used in this project?
So as we said in the methodology, that first we do a recombinant DNA technology, and also we will detect microplastic presence in our samples. First, materials that will be used in recombinant DNA technology, it's first in plasmid construction we use PET11A CYP119 from Adgene, which is commercially available and which has been codon optimized in E. coli. And in gene cloning and bacterial transformation, we use E. coli cells DH5 alpha and an ampicillin antibiotic. In gene expression induction, we will use 0.1 micromole of IPTG, which will act as an inducer of protein expression, and also we'll use a gel electrophoresis method for the detection of transformed cells.
So now, how will the proteins that we will use be purified?
Now, after the expression, two proteins will be produced, PETase enzyme and GFP. The cell will first be lysed, and after lysis, the debris will be washed, leaving only the chimeric protein in the sample. And we will use mass spectrophotometry and SDS page sodium dodecyl sulfate polyacrylamide gel electrophoresis to confirm the presence of PETase GFP protein, as it will be confirmed by the presence of bands on this gel electrophoresis. And now, after confirmation of these proteins, we will expose the sample to the nickel matrix, which will unbind the histag. Histag actually is the combination of histidine proteins, which now help in efficient isolation and purification of proteins from the sample. Now that we have made our bioreporter, which is made of GFP peatase enzyme complex in an E. coli cell, now Nadine, how will we test that our bioreporter formed is efficient on microplastics?
Yes, so since it will work on microplastics, as you said, we have to first prepare the microplastic. So we will have to purchase it from a manufacturer. We know that we say that it comes from a plastic, a usual plastic. So we will buy a PET plastic, and then this plastic will be crushed at a low temperature to achieve a size that is less than 100 micrometers, or the usual size, which is 50 micrometers. So, and then we will have to do a microscopic examination to observe the level of fluorescence. Now, so after that, we will expect to observe the fluorescence protein, the green fluorescence, which will come from the bioreporter that we have engineered. Since the samples, we know that they contain microplastics, and so that is what we will observe. So now, can you tell us in summary what the project will be talking about?
So our project, as we said, it's on making a bioreporter which is efficient in detection of microplastics in aquatic environment, and we said that our project is crucial because current methods are labor intensive and costly and requires a skilled labor, while our method is performed on site, and it is for a short period of time and also cost effective. We aim to engineer an E. coli bacteria to produce a PETase GFP complex or fusion protein that will fluoresce in the presence of microplastic in the aquatic environment. Now, early detection and monitoring of microplastic pollution is crucial for environmental protection and health management. Thank you all.
Thank you.